And our next speaker today is Dr. Michael Reimann, Group Leader for Connectomics within the Simulation Neuroscience Division of the Blue Brain Project. Michael.
Hi, thank you. Thank you for letting me present my work here. And I must say, after all these great presenters, I feel a little bit inadequate. Okay, so I'm from Blue Brain at the EPFL in Switzerland, and we are also doing detailed modeling of neural circuits, similar to the model that has been presented before, the V1 model. We are modeling detailed models of neural microcircuits, be it cortical circuits or hippocampal circuits. We model them and we simulate them. And we do this in order to address questions that are challenging to be answered with purely experimental methods or with more simplified models. Just as a motivating example, it is known that in neural circuits there are several sources of noise. For example, the chemical synapses connecting the neurons are actually very, very unreliable. And so there's this uncertainty. And through the recurrent connectivity in a network, this uncertainty might get amplified, leading to the question, how can these circuits actually perform any computations and encode anything? To what degree is the neural code limited by the internally generated noise? And in simulations, we can address this very simply in a model by simulating it up to a point, freezing it, and then resuming from that point in time, but with different outcomes for the stochastic processes that explicitly simulate all these sources of noise. So we get two alternative realities that are both equally valid. And then we can study how quickly they diverge from that point. And since we control all the noise sources, we can selectively enable them or disable them and see how that affects the results. And this study is currently in press, and it's a nice study, if I may say so myself. However, it was to some degree limited in that we only study the dynamics of a tiny little part of the brain, and we had to assume that all the external inputs that this is receiving, which is like 80% of all the inputs, are just a constant external input, just a constant signal, which is not how the brain works. Everything is connected recurrently. There are way more complicated dynamics. And so to address that, we have to scale up, have to go to larger models. Eventually, we built a model of whole neocortex of a mouse. And in doing this, you run into the problem that if you want to do detailed modeling of a whole neocortex, you have to connect the neurons. And since, as I said, it's detailed modeling, you have to connect them on a subcellular scale. So you have to know the exact locations of all the synapses. Not only the exact neurons they target, but also the exact location on the neurons. And not much is known. Already on the local scale, not much is known. If you go long range, even less is known. Well, some stuff is known. There is, on the macro scale, if you look at connectivity between brain regions or between voxels of the brain, there's actually good solid data available, but that lacks the cellular resolution. And if you go to the micro scale, connectomic studies on that level tell you that there is non-random structure which is likely to be very important, so quite the dilemma. And our approach for this dilemma was to ask the question, the data on the macro scale, what micro scale does it actually imply? If we just take all the data we have on the macro scale, apply all these constraints, what kind of micro scale connectivity do we get? Maybe it's better than we think. Just as an example, if we start with a region-to-region connection matrix, which is giving us the strength of connections between individual brain regions, what is the most general model of the microstructure implied by this? Well, here's an example. The strength of the connection gives you an idea how many connections there are anatomically. And then the most general model would be to have all pairs of neurons in these two regions connected with a uniform independent connection probability. And that probability is given by the strength. That would be the most general model so far. But we know more. There are more constraints. For example, layer profiles. So based on the data that the Allen Institute published and provided, we predict them for all of the projections in the mouse neocortex, a certain layer profile of synapse density in the target region. So where in the region along one dimension all of the synapses are actually located. And that already constrains which neurons can be targeted because they need to have some morphology in those layers. That's one constraint. Another one is the mapping of connectivity, which we assume to be topographical. We assume, so the idea is that you have connectivity region to region, but it's not uniform. You have certain parts of a region connecting predominantly to a certain part of another region. And in that, there is a certain type of topographical mapping which can look like this. But it can also be less precise, can be more precise, less precise. It can be partial, that only a part of a region is mapped to another region. It can be inverted, or anything else. How do we parameterize this? We parameterize this using a voxelized mouse brain connectivity model also provided by the Allen Institute. So this provides higher resolution than region to region. And then we parameterize the mapping in the connectivity between regions the following way. We first define a local coordinate system in the source region, in this example, primary visual, but it could be any. This is just an example. We define a local coordinate system here by placing 3 points in it. That defines local coordinate system. And here, for visual purposes, I color these 3 corners red, green, and blue. And then we can look up in the voxelized connectivity model the connections in this blue corner. Where are they going to? And color everything that they innervate in blue. Red corner, red, green corner, green. If we do this, excuse me. If we do this, what we get looks like this. So here is the source, and we see innervation of all the surrounding visual regions. And we see that there indeed is a certain structure revealed. We see that certain parts of these regions are innervated predominantly by certain corners. And we can parameterize this fact by also defining local coordinate systems in all the target regions by placing 3 points, coloring according to the same coloring rules as in the source region, and then optimizing the location of the 3 points so that the coloring scheme given by the data is reproduced. Now, that defines a mapping in that you assume red maps to red, green to green, and blue to blue. Now we validate this then against independent data. And it's not just independent data, it's totally different data in that this is not anatomical data that's telling you what is wired where, but this is functional data telling you that this part of V1 of that region, what part of the visual field it represents. So what part of the visual field do you have to place a stimulus in to activate this part of the brain? And then we simply assume that if there are 2 parts that are representing the same part of the visual field, then they are wired together. That's why they represent the same part of the visual field. They are connected to each other. And when you do this kind of stuff, you see that for some parts of the mapping, there is a reflection operation involved. For example, from here to here, you see that what's counterclockwise turns clockwise. And in fact, in this paper, this fact, it's also from the Allen Institute, I think, this fact is used to find the borders between the various visual regions. And if we compare all of the presence or not of a reflection operation and rotations in the mapping to our prediction based on this anatomical data, it's a good match. So that's another constraint. One last constraint we applied, what we call projection types. So there are certain projection classes known, but even if we just take one projection class, if we just look at the intratelencephalic projections from a somatosensory region here, this part, it innervates many other brain regions, but what does that mean really for an individual neuron that is located here and is sending its axon out? It could be that individual axons are each of them innervating everything. Or it could be that they split duty and just one axon is here, one is here, one is here. Or it could be something random, some random process. How do we model this? Well, for that, we, as data parasites, go to another openly available data set, in this case, the Janelia mouse slide data set, which is actual reconstructions, highly detailed reconstructions of long-range axons, individual axons. And so we take these axons, and for each of them, we analyze which brain regions they are actually innervating. For example, here for 39 axons from a motor region. And, well, we see that some innervate more, some innervate fewer, and that some brain regions are more likely to be innervated and others are less likely to be innervated. So there are these various first-order innervation probabilities. If we calculate these probabilities and compare them to the strength of a projection, so that's a predicted innervation probability, essentially taking the average along this dimension, compared to a prediction based on the projection strength in a macroscopic connectivity model, we see that one can predict the other, which is not very surprising. It simply tells you that a projection is strong because many neurons participate in it, and only to a lesser degree because individual neurons project strongly. So we can predict this now, but what about the higher level structure of this? They're clearly, it is not clearly just not just the first order innervation probability, there are statistical dependencies. Well, what we do for this is we calculate for pairs of potentially innervated brain region the statistical dependencies as the change in conditional innervation probability. If you know that a brain region, B in this case, is innervated, what is the probability that the brain region A is also innervated? Divide this by the first order innervation probability. How does it change? We calculated that for all of the data shown here, and indeed for many pairs of brain regions we found that there is a clear statistical dependency. In fact, what we found is that for many pairs of brain regions there is this dependency, and it is always an increase in probability. So, for example, it's never the fact that there are two brain regions where the innervation is mutually exclusive. And this fact we try to model with a tree-based model. Here I'm just illustrating the principle. Imagine you want to model what the axon of a single neuron in this region, MOS, is innervating. Well, you just move along the tree towards the right. Each edge can be crossed with a certain probability or not. And then you cross, and if you decide that you cross all of these edges reaching a leaf, then you assume that this is innervated. And that explains, in this model, that explains the observed positive statistical interaction in that when you know that this is innervated, then in order to innervate this region, you only have to cross one more edge. For this model, we get the structure of the tree by clustering the macro-scale connectivity matrix. and can generate with that predictions for the innervation of all the regions, again, along this axis, of individual axons in, well, all of the brain regions that compare pretty well to the data. In fact, we can show that if we calculate the pairwise Hamming distance of these profiles, we can show that in the data there is a very wide distribution that's better modeled by our approach than by the naive model that only takes into account the first order innervation probabilities and not the statistical interactions. We can also validate it against an independent data set, and in this case, a data set in a totally different region. What I showed you so far, the axons that we used were in a motor region, but there's also data available in a visual region here. In this case, Han et al. Looking at innervation of single axons in layer 2, 3 of primary visual area, the number of other visual areas that are innervated, and the statistical interactions, which our model, well, there's a qualitative match. Now, with all of this, these are very simple constraints. It boils down to it. It's very simple constraints based on macroscopic data. We now generate an algorithm that is actually generating instances of micro-connectivity between 11 million neurons in a modeled mouse neocortex, placing around 85 billion synapses. But these statistical instances fulfill all of the constraints that I just described. What we can then do is ask the question, well, what kind of microstructure do we get? We now do micro-connectomics on this model. Now, one thing that's typically done in micro-connectomics is looking at, for example, reciprocal connection probability. It's been a pretty robust finding that if you have a connection one direction, you tend to have a connection in another direction. So we're sampling pairs of neurons that are now in different regions. And we find that indeed, between these, there is an overexpression of reciprocal connection probability, but you have to sample the neurons over a sufficiently large radius. It's our prediction. And another thing that's typically done in microconnectivity analysis is looking at neuron triplets and which types of motifs are overexpressed. So you sample 3 neurons, each of them in a different region. And there are 6 potential connections. That leads to 2 to the 6 equals 64 potential motifs that we can have here along this axis. And you see how many you find when sampling compared to an expectation, a null model based just on connection probabilities. And indeed, in this still very simple model, we find that there are certain motifs that are significantly overexpressed. And I think that demonstrates a little bit the power of this as a null model, in that if somebody did this actual experiment and has this finding, they might be tempted to hypothesize that this is a very specific mechanism. But I show that this can be explained very simply by very simple principles, by layer profiles, topographical mapping, and these non-random projection types. And of course, with these instances, we can also now simulate potentially whole neocortex, not just us, but also other groups, because we make this available for download, both the parameterized constraints and the instances. And then with this, and thanks to my co-authors, I'm done.
Wonderful. We have time for a question or two. Christoph?
Michael, how do you deal with distributing the, what constraint do you use to put synapses onto the dendritic tree, spine, soma, etc.? What rules do you use?
Right. So we know the number of synapses that we have for a given projection based on the strength of the projection. We know the layer profile. And then we simply, we discretize that layer profile in bins of 100 micrometer. And then we simply pick random dendritic segments that are in that part. So what I have not mentioned is that before all of this, we actually have tons and tons and tons of neuron reconstructions that are placed in a model of mouse neocortex according to our best idea of what the actual composition is. Composition of pyramidal cells, various interneuron types, and so on and so on. We place them so we know how much, for example, L5PC dendrite is in layer 2 of that in that brain region.
So you do have
and then we pick, yes,
specificity in there.
Yes, I mean, we have cell type specificity in terms of the composition, but then what we then assume is we assume nothing in terms of targeting specificity that goes beyond that. We assume if you have a dendrite in layer 1, 2, 3, and according to the layer profile of synapse density, layer 1, 2, 3 are targeted, you are eligible for being targeted. We do not specifically target only L5PCs or only interneurons or anything. If we find good data, we could add that. So, I mean, that's the idea. It's a continuously evolving model.
Okay. Thank you very much.